These are the lecture notes for the extratubular digestive system. And I've gone through them and I've highlighted things that I want to draw your attention to, okay? And then a lot of these things that I've highlighted to draw your attention to, I'm going to actually show that to you on the screen with the slide. So just to get started, we're the extratubular digestive system. We need to be thinking about glandular epithelium. So the smaller glands, the mucosal glands that we've looked at, the gastric glands, the intestinal glands, they were formed from the lamina epithelialis. So those are epithelial glands, but they're small. They're just invaginations of that lamina epithelialis down into the tunica mucosa. Now we're talking about a very large mass of epithelial cells that was so big when it invaginated down from the lamina epithelialis, it kept on going. It went through all the tunics, it came out of the digestive tube and then formed out in the connective tissue outside of the tube. So that's why it's called extratubular. Now, these epithelial glands are so large that now they have to have a duct system to help them get their secretion back into the lumen from which the whole gland originated. And not only do they have to have a duct, but they have to have a duct that branches to get to all of the little secretory units within that gland. That branching word, we use compound. The word compound means branching ducts, because branching, that word was already taken for some glands, simple glands that branch. So we couldn't say branching, so we had to come up with another word for branching, and that word is compound. So ducts that branch means it's a compound gland. If the adenomere branches, then we just call that branching, okay? And that's referring to just the adenomere. Now, the adenomere is the functional unit of the gland. I've got that highlighted here. So the secretory product in these glands is made by the secretory units, and we call those secretory units adenomeres, and those are the functional units. Now, in addition, we have the stroma, the connective tissue framework that supports and surrounds and shapes that gland. Now, there's another term, parenchyma, and parenchyma is kind of everything outside of the connective tissue stroma. So parenchyma includes the adenomeres and the ducts. So when you look at a gland, I'm going to zoom back out, okay? You'll see that the gland, it has a capsule, and we'll see that in gross anatomy. So you'll dissect that capsule, or you'll dissect the connective tissue away from the capsule of that gland. You've seen the lobes of the pancreas. Those are encapsulated, and it just so happens that the capsule of the pancreas is kind of clear. So you can look through it and you can see all these tiny little lobules in the lobes of the pancreas. And I really want you to see that next time you're in gross anatomy. Those tiny little lobules are surrounded by connective tissue septa. So the capsule, the septa that surround the lobules, and any built up connective tissue around the blood vessels, the autonomic ganglia, the duct system, the interlobular duct system, all of that is your stroma. That's your connective tissue. Parenchyma is everything else. So it's everything inside these lobules. And what is inside these lobules? Well, the adenomeres, the functional units, these guys right here, and then the ducts. And the ducts are ducts plus adenomeres equals parenchyma. There, I love to make those little math formulas. It helps me remember things. Now, moving down to general classification scheme for exocrine glands. This is kind of coming a little early in the course. We've got exocrine glands and we've got endocrine glands. And exocrine glands, their mode of secretion, there's 4 subtypes: merocrine, apocrine, holocrine, cytocrine. These are things that you're not really going to get to see, taste, and smell, so to speak, until we go into the integumentary system, which is the second to last system in the course. So kind of put this on hold, and we'll come back to this, and we'll think about these definitions and how to apply them to actual examples in the integumentary system. Moving on down to the number of cells in a gland, we've met the unicellular glands, the goblet cells. Now we are meeting the multicellular glands. And multicellular glands can again be endocrine, which don't have ducts. That is the last organ system in the course, so put that thought on hold. And then the exocrine glands, which often have ducts. Some of these exocrine glands are simple and some are compound. Simple glands either deliver their secretory product directly into the lumen, like the gastric glands did, or the intestinal glands. Or they may use one duct, but it won't be a branching duct. And some of the submucosal glands we saw both in the esophagus and in the duodenum, they used a duct sometimes. Sometimes they just dumped their secretory product into the fundus of the intestinal gland. But those were simple glands that either don't have a duct or they have one duct and that duct doesn't branch. Today, we're going to look at compound glands, and they're so big and so complex that they have to have branching ducts to get to every little adenomere, every little secretory unit, and drain all of those. And all those ducts will converge together to form the major ducts of the gland. Now, you've already seen down in anatomy that glands have lobes that are subdivided into lobules. I gave you the pancreas as an example. Even the liver has lobes. And then the lobules, you can't see without a microscope in the liver, but in the pancreas, you can see the lobules down in the gross anatomy lab without a microscope. Okay, so we're looking at compound glands today, and that means their ducts branch, okay? Well, these ducts have names as they start out very small ducts with a very small diameter to their lumen, and they converge together and form larger ducts. They have this sequence as they transition, a sequence of names that we use for these ducts. The smallest beginning of the duct is intercalated, an intercalated duct, and that is a subtype of an intralobular duct. So we're inside the lobule, and inside that lobule, we're going to have two intralobular ducts. The first little portion is going to be called intercalated. The next portion will be called intralobular. We know that the intercalated is also intralobular, but it's just the way the terminology is applied. So we'll start out with intercalated, and then we'll go to intralobular. Both of those are inside the lobule. Then we'll move outside the lobule, and we'll look at inter, I-N-T-E-R, lobular. Now, the bigger lobar ducts and the main ducts, we won't see. For example, we won't see on the microscope. You'll see that in gross anatomy. Some of you had livers where we couldn't really see the individual hepatic ducts that were draining bile from each lobe of the liver. Some of you had livers that it showed up really well. So those were big lobar ducts that drained each lobe, and those were called hepatic ducts. And then they converged together, remember, to form, with the cystic duct of the gallbladder, they formed the main bile duct, okay? So those were what we saw in gross anatomy. So what we're seeing in histology today is two subtypes of intralobular and then the interlobular, which vary in size. They'll start very small and then they'll get larger. All right, so let's take a look at those ducts. So here we are back at this salivary gland, okay? And let's see, we'll zoom in right here because I found some nice examples. Here we are zooming in on a lobule. Okay, and we're looking at just the ducts. So this is a mucus secreting salivary gland, okay, and we'll take a look at that and how we know that in just a minute. Let's just think about the ducts right now. So when you look on low power mag, you can see all these ducts scattered all over the place. This is a salivary gland, so a lot of these ducts are very acidophilic in nature, okay. So, but mostly we see the adenomeres, but we do see a pretty good bit of ducts in all kinds of different profiles. They may be in longitudinal profile, they may be in cross-sectional or transverse profile, they may be oblique. So we may see their lumen or we might not, depending on how the knife blade went through that duct. This is transverse, this is more longitudinal. Now, right here is the beginning of the duct. These are the little adenomers, and their secretory product is delivered here into this little tiny beginning. This is called an intercalated duct. It's so small that that's why it's called intercalated. If you are intercalated, you're kind of inserted between two things. You're inserted here between the adenomere and the striated intralobular duct. So it's a tiny little intercalated duct, just not much to it at all. Small lumen, very short in length. Intercalated ducts right there. And depending on how the knife caught them, because they're so tiny, they'll have all kinds of different little profiles and appearances. But again, they'll be very, very short and small in diameter. So those are your little intercalated, which is a subtype of intralobular, but we're going to call it intercalated. Then the next intralobular duct is this larger duct. And in the salivary gland, these are called striated ducts. Why are they called that? Because they have all these invaginations on the basal portion of their cell membrane, and all of these little ribbon-like enfoldings are where they store mitochondria. So all these acidophilic staining regions right here are shelves of cytoplasm filled with mitochondria. What does mitochondria mean to you? Active transport. So these ducts are actually contributing to the secretory product. They're transporting ions, they're secreting like bicarbonates into the lumen to join with the secretory product that was released from the adenomere into that lumen, now mixed together. So those are the two subtypes of intralobular ducts in the salivary gland. Okay. Now, I-N-T-E-R, will look between the lobules, and we're going to look for more connective tissue. So in an area like this that has a lot of connective tissue, we're going to expect to see nerve fascicles, we're going to expect to see autonomic ganglia, blood vessels, arteries, veins, and this is a duct. Now these ducts tend to have, they don't always have, but they tend to have a striated epithelium, stratified epithelium. They don't have to, it may be a simple epithelium, but they tend to have a stratified epithelium. This epithelium is stratified right here. We've got a row of nuclei down here along the basement membrane, and then we have another row of nuclei here on the apical compartment, okay? So these are actually, this is a bilayered epithelium right here, so it's stratified, and you name it by the shape of the more superficial cell. So these are columnar, so this is stratified columnar. Sometimes you'll see cuboidal mixed in, so I usually like to say stratified cuboidal / columnar because it kind of varies. Now, I don't know why, but the more basal layer of the stratified duct epithelium, those nuclei will never appear as regularly as the more superficial layer of cells. And I don't know the reason behind that, but that is the way it is. So don't expect a double regular row of nuclei. Expect that basal compartment row of nuclei to be more spotty, more irregular. Okay. So that's your big interlobular duct, okay? And then we won't see the lobar ducts and the main ducts. You'll see that down in gross anatomy. Okay, now let's talk about the shape of the adenomere. In the case of the intestinal glands and the gastric glands, remember in the large intestine, those were simple straight tubular glands. So the large intestinal glands were simple straight tubular. The gastric glands were simple tubular also, but they had some coiling and they might even have had a little branching. Okay, but those are still simple. They emptied out directly into the lumen of the digestive tract. So these are examples of simple glands right here. Simple straight, simple branching, simple coiled, and they're tubular. Today we're going to be looking at compound glands, and these compound glands, they'll still have these they can still have similar shapes to their secretory units, but they'll be stuck on the ends of the ducts that branch. So for example, what we'll see today, this is representing a duct and it's branching here and here and here. Then we've got a tubular portion and an acinar. And acinus means round, acinar means round. So tubulo-acinar. And compound because the ducts branch. That's what all the glands that we're going to see today, as far as salivary glands and pancreas are concerned, they are compound, then tubulo-acinar. So compound means the ducts branch, and then the adenomere shape is tubular and acinar. Compound tubulo-acinar glands. Okay. Now, what about the secretory product? Well, it's fixed in the pancreas, but in the salivary glands, there can be different flavors of salivary glands. There can be mucus-secreting salivary glands. There can be serous-secreting salivary glands. There can be seromucus or mixed types of salivary glands. Mucus, the cytoplasm of the cells will have mucin granules. Serous, it'll be basophilic staining, usually, and often has zymogen granules. Remember, that's just a generic name for proenzymes that are packaged and waiting to be released. Now, seromucus or mixed, they can go about how they get to be mixed in different manners. The example you'll see today is something called seromucus with serous demilunes. That means the adenomere itself will be the tubular portion, and kind of the beginning of the acinus will be mucus secreting. Then it will be capped with a baseball cap here, or a half moon. That's why it's called a demilune. Demilune, demi means half, or petite moon, a half moon of serous secreting cells. Or we may see a purely serous acinus, or we may see a purely mucus acinus. Okay, well, what does that look like in reality on the slides? Well, this slide I've already got up here is a purely mucus secreting salivary gland. Let's get in a not blurry place. You can see some of the tubular portions of the adenomeres. Then it'll end with a little rounded acinus. Okay, you can see little portions of the lumen of these adenomeres. Here's a longitudinal profile. Here's transverse, where you just see kind of a little circle in the middle. Okay, so these are the lumens. These are the acinar cells. Each individual epithelial cell is called an acinar cell, A-C-I-N-A-R. These are mucin granules. So these are prepackaged granules that are waiting to be released. And the secretory product combined with sloughed epithelium and a little bit of fluid, it will be called mucus. Okay, mucus, but these are mucin granules. Okay. And you can see that this acinar cell is so full of mucin granules, his poor little nucleus is just squished down here in the basal compartment of the cell. All right. So this is mucus secreting salivary gland. But what else do we have? We have a seromucus salivary gland. So let's zoom in on that. Okay. He's still got the pretty little striated ducts here with that acidophilic kind of, you know, scratchy looking. You see all these little lines here where all those mitochondria are stacked up on shelves. So those are striated ducts. Those are the stria or striations that you see here, all those little lines. Now I've zoomed in here and you can see all these mucin granules. So you're thinking, okay, this is a mucus secreting salivary gland, but wait, what are these guys right here? They're just slightly more basophilic. Better pay attention to those. Let's zoom back out a little bit so we're going to get a more holistic view of it right here. Here's a little intercalated duct right here, sitting off by itself. And then here are striated ducts right here. But see, we're consistently seeing these little end pieces, little collections. Here's a demilune right here. You see the little half moon? Okay, so we've got serous demilunes here. So the serous portion is being secreted into the lumen of this adenomere right here, and then these mucin granules are being released right here. Now this secretory product is going to enter right here, just right there into that intercalated duct, this little short connecting piece right here. That's why it's named intercalated. And then that will empty into the lumen of the striated duct. Then the secretory product will flow into the interlobular ducts. So here's one here. And that is another pretty stratified cuboidal to columnar epithelium, INTER. And these guys just get bigger and bigger as they head out toward the main lobar ducts. Here's another area where on low power mag, we can see those real pretty serous demilunes. So this is a seromucus secreting salivary gland. Last but not least, if you like red, you're going to love the serous salivary gland. Here is one right here. And they are real eye-popping because their cytoplasm is filled with zymogen granules. And these are proenzymes that are packaged and waiting to be released. So yes, digestion does start with the addition of saliva in the oral cavity when we're chewing. Now, sometimes the cells will be kind of empty, and you can see that they have a basophilic cytoplasm in the background behind all of those red granules. That's because these are zymogen granules. These are proenzymes. These are proteins. So these are protein-making machines. So of course you expect their cytoplasm to be basophilic because it's just loaded with RNA, okay? But you don't always get to see that because if they're chock full of these acidophilic zymogen granules, then you don't get to see the really pretty basophilic cytoplasm. So here's one acinar cell here, here's his nucleus, and there's his cytoplasm there. Now this is a striated duct right here. The intercalated ducts look a little different in the serous salivary gland. There's a little bit of one right here. Let's just drive around and see if we can find more. Here's a little tiny intercalated duct right here. Just look really different in the serous salivary gland. They don't just pop out at you like in the other glands. You have to kind of drive around and let your eyes adjust. Here's a tiny little intercalated duct right here. And then this is the striated duct. I don't know why, but I've never had a serous salivary gland scan in good. I think that all these refractile granules here just kind of irritate the scanner, and you don't get a real good strong resolution of these granules. It'll always look a little glowy and murky, but that's just the serous salivary gland. Here's an intercalated duct right here. Here's one right here, okay? And those are emptying into these striated ducts right here. And this is a striated duct that just didn't have good preservation, but this one preserved very well. Okay, so that's the three types of salivary glands. All right, now, do you all want me to carry on to the pancreas, or do you want me to stop and take questions on the salivary glands?
Carry on.
Okay.
I think you have a question in the chat.
Okay. We have a chat question. Let me run over.
Yes, we do. It says, if you are not in the salivary gland, would the flow of the secretory product be intercalated, intralobular, instead of striated, and then the interlobular duct? Good question.
Very good question. You want to answer that, or you want me to?
Go ahead, you're on a roll.
Okay, I'm on a roll. All right, so if you're in the salivary gland, here's the flow of saliva. Adenomere, intercalated duct, striated duct, then interlobular, and then that's all we're going to see on the microscope. But if you're in the pancreas, it's a little different story. Okay, are you ready for that story? All right.
Yeah.
That's a perfect lead into the pancreas.
Yeah, it's a great question. Boy, it's almost like we planted it, you know? All right, so here we are with the pancreas. Now, the pancreas has an endocrine portion, pancreatic islets. We'll talk about that in the endocrine system, the last organ system of the course. And then it has an exocrine portion. Grossly, you saw that it has the body, right and left lobes. Microscopically, it has lobules, and it's also compound tubuloacinar. Now, the tubular portion of the tubuloacinar is not very prominent in the pancreas. And the salivary glands, especially the mucous salivary glands, you can really see the tubules better than the acini. But in the pancreas, it's the opposite. You see the acini better than the tubules. Go figure. That's just biology for you, biological system. All right, so adenomeres are, again, tubuloacinar, but you're going to get more of the acinar in the pancreas. We call each individual cell an acinar cell in the pancreas, just like we do in the salivary glands. So we have pancreatic acinar cells. They have spherical nuclei that are really euchromatic. They have extremely basophilic cytoplasm. Again, they are protein-making machines. They have intracytoplasmic proenzyme granules, which generically we will call them zymogen granules. And then they have a little extra spice to their ducts. They're going to push some of that intercalated duct, some of that epithelium up into the lumen of the adenomere and start that duct early. They just want to get a jump start on it. And we're going to call those centroacinar cells because they're in the center of the acinus, okay? And that's the beginning of their duct system. Their duct system will be centroacinar cell, intercalated duct, intralobular duct, and then INTER lobular duct. So their intralobular duct, we won't call it striated because it isn't. They don't have all those stria, all those basal infoldings of the cytoplasm, and they don't stack all the mitochondria in their ducts, okay? In fact, ducts are hard to identify in the pancreas, but we'll take a look at it. So this is just showing that the central acinar cells are kind of evaginated up into that lumen of the acinus. Then you'll have your little intercalated, then you'll have intralobular, then you'll have interlobular. And this is just a schematic showing you all the little tiny adenomeres, and they're emptying into that tiny little intercalated duct right there. And then those intercalated ducts empty into the intralobular ducts, and those intralobular ducts converge and converge and converge, and then exit one lobule right here. See the septa going around that? They exit that lobule and become INTER, interlobular ducts. Then the big lobar and main ducts we won't see in the microscope. We'll see those in gross anatomy. Okay, so let's take a look at that pancreas, and that is this slide here. Okay, so even at this low power mag, and we went over this slide in our last lab, but we went through it pretty quick, so we're going to do it again. Here's the artery supplying it. Here's a vein draining the pancreas, and we'll look at those branches supplying the pancreas in gross anatomy. Here is a big INTER, interlobular duct. Here's a smaller interlobular duct. Smaller yet interlobular duct. So it's going to start small, get a little bit bigger, get a little bit bigger, and just keep converging and getting larger until you have those bigger ducts that we see down in the gross lab. Right here, we've got a couple of INTER, interlobular ducts. We're going to zoom in and we're going to try to find some intra. Here is an intralobular duct right here. What drives me crazy about the pancreas is these ducts don't have a lot of connective tissue around them, and they're hard to ID in the pancreas. Just the devil to ID. But what you want to do is you want to relax about that, because I'm going to ID them for you and take pictures for you in the exam. So you just got to figure out the flow. And here's the key. The smallest ones are the intercalated. Okay, so let's zoom back out. That's kind of small right there. I had a favorite. Here we go. All right. So here's a longitudinally profiled intralobular duct. That's about as fancy as it gets for an intralobular duct in the pancreas. And then here's a teeny tiny little intercalated. And this intercalated, the entire diameter of that duct is about the diameter of one acinar cell. So here's an acinar cell right here, and there's that intercalated duct. So it's all about size. So if you see a duct that, you know, its diameter is about the size of an acinar cell, boy, that's intercalated. That's tiny. Next will be intralobular, which isn't a heck of a lot bigger. Some of them get a little bit larger than that. This is intralobular streaking along right here. This is the endocrine portion. This is a pancreatic islet. We're not worried about that. This is an intralobular duct islet. This looks like, gosh, not sure about that one right there. Run away. All right, here's a blood vessel there. Okay, what else we got going on here? blood vessel. Blood vessel, here's a tiny little intercalated right here. Okay, so that's the smallest of ducts right there. Now let's take a look at the acinus. Here is one acinus right here. You can see some connective tissue around that adenomere. See how it's kind of round. Sometimes they're a little tubular. So here's your tubular portion, here's your round portion, tubuloacinar. Right in the middle, shining like a diamond, is the central acinar cell. And once your eyes get used to it, man, they really start popping out like stars in the sky. There's an central acinar cell. Here's a lovely one right here. Okay, that's the beginning of the intercalated duct where it's evaginated up into the lumen of that acinus. So central acinar cell, then intercalated duct, then intralobular duct. Where do they go? Intralobular duct. Oh, here's a nice one. This one's getting, there's an intralobular duct right here. This one's almost ready to be an INTER lobular duct because it's starting to get a lot of connective tissue, but it's still in the lobule. So we'll call that one intralobular, just a large intralobular, average size intralobular. Here's the lumen of an acinus and longitudinal profile. And wherever you see those shiny diamonds like this right here, that's a central acinar cell. Now, here you can see one acinar cell right here, just the most incredibly purplish colored basophilic cytoplasm from all of that RNA, making all that protein, very euchromatic nucleus, and those apically stored zymogen granules. Remember, these same red or acidophilic, grainy looking granules, zymogen granules, will also are in the serous salivary gland because the serous salivary gland is responsible for starting digestion in the oral cavity. Here's some central acinar cells right here getting ready to start an intercalated duct. Okay, I think that's all we need to see on the pancreas. Any questions about that, dude? Oh, one more thing is, I don't know why, but in the pancreas, the epithelium tends to be more simple in the INTER, the interlobular ducts. And it is not a pretty stratified epithelium like in the salivary gland. Don't know why, that's just the way it is. Yeah, that's all I got to say on the pancreas.
Okay.
Any
Carla,
questions on that?
yeah, we have a chat.
Okay.
And in the chat, it says, I'm confused by what acinus means in terms of cell structure. How does it relate to adenomeres?
Oh, okay. Okay, good question. That is a source of confusion for beginners and sometimes for people who've been doing it a while, right? Okay.
I agree.
So adenomere is the secretory unit, and that adenomere can be in different shapes. And Tom, I might have to have you read me that question again so that I
Sure.
don't blow it. Will you read it one more time?
It says, I'm confused by what acinus means in terms of cell structure. How does it relate to adenomeres?
Oh, okay. Yeah. Okay. So an adenomere is the secretory unit, and they can be many different shapes. This one is tubuloacinar. An acinus is round. Okay. So I used to tell students in the past, tubuloacinar is like the long green grapes you can get at the grocery store. And then you stick a little round red one on the end. And then you've got a tubuloacinar adenomere. The whole thing is the functional unit because the whole thing is creating the secretory product of that gland. The acinus is just the round red grape stuck on the end. We happen to call all of the cells in that adenomere, though, we'll call them all acinar cells, not just the ones that are in the round part, because acinus tends to mean round, but we'll also call these acinar cells that line the tubular portion. Go figure, it's just the way it is. Okay, did that answer the question?
That answered the question perfect.
Yep, thank you so much.
Okay.
So when it comes down to it, acinar is describing a shape.
Right, and we just call the epithelial cells that line those tubuloacinar secretory units, we just call them acinar cells. Okay, so take a deep breath. we've made it through salivary glands, the pancreas. We've got the liver and gallbladder left. Okay, so we're gonna need to shift gears a little bit. We need to kind of think a little differently, okay? And I'm gonna jump around on slides a lot because each liver slide has something to offer that the other doesn't have to offer, okay?
Hey, Dr. J, can I snag you really quick? I don't know if it's the same for everybody else, but when you are like clicking through the slides quickly and like moving around and you're like, okay, so here's this, like your mouse is moving at the same speed that you are, but the slide is like lagging way behind. I don't know if that's the same issue for other people, but it'll say like, here's blood vessel, and you're circling something random, and then it comes in and pauses where you were just circling. So if you can like take a half an extra second for the slides to catch up when you're showing us certain things.
Oh, wow. I didn't know that was happening. Thank you so much for letting me know.
Yeah, it's not a huge deal, but if it would help if it's just a half a second.
I think
It is a huge deal.
and I think it's a great challenge that we have because I'm not having any of that delay because I'm in the building with Carla. I don't know students that are, it's that I believe that comes down to when I've spoke to IT about that, that may have to do with the capability of Zoom if you're an off-site space. So in other words, for me, Carla, I'm seeing it same real time you are, but if I was at home or in an apartment, it may be a little different. So I think it would be smart just to move a little, pretend you got proto slow and kind of move a little slow across the slide because it's going to be extremely variable depending upon where your internet is.
Yeah, thanks for telling me that. You know,
thank you though. Great comment.
We're usually in the vet lab and you're all in there at one time.
I'm in the student lobby and it's still chunky today. So yeah, I don't know.
It's good for us to know because if somebody doesn't say something, we don't know what's happening if we don't experience it.
Yeah, please shout out something like that.
That's great. So going slow may help. And if it doesn't get any better, please remind us.
Yeah, or if I forget, yell at me. It's really okay. You can say, slow down. And I'll be like, oh God, yeah, okay. All right, so here we go. Liver, largest gland in the body. I think we saw that already in the anatomy lab, right? Boy, those things were huge. Okay, so they have a stroma, have a really thin connective tissue capsule. So if you know, that's just the visceral peritoneum, and it's really thin. Now, that capsule will go on into the parenchyma or the bulk of each lobe, and it sends septa down in there, and those septa will surround the lobules in some species. Okay, hold on to that thought. So the liver has lobules, it has lobes. You saw the lobes, right? Quadrate, right and left medial, right and left lateral, caudate with all its funky little processes. So you know it has lobes. Now you're going to find out that it has lobules. Those are microscopic, and only the pig, thank God for the pig, put connective tissue around each lobule so that we can see the darn things. Okay, so just to start out with respect for the pig. I mean, we laughed at him about his lymph nodes. Now we got to tell him we're sorry because he's saving our butts here on the liver. So here is a pig liver, and you can see all these discrete lobules. And this is the only species that is going to grace us with these little fine connective tissue partitions around each of the lobules. Okay, so that's just wonderful because that's going to help us learn about the liver today. Okay, so lobules. Now, what do we need to look at inside the lobules? Well, let's see. We've got parenchyma, vasculature. I think I want to start with the vasculature because if you don't understand the vasculature, then you don't really understand how the liver works. It's kind of like the spleen, okay? So you need to understand how that blood is flowing in. Now, unfortunately, that's a little premature for the anatomy lab. You had to cut. When you cut the liver out, if you looked at the area where you cut in the hepatoduodenal ligament there, you cut the portal vein. You actually cut hepatic arteries. You might have seen the green latex, and you cut the bile duct. Okay, So you would have that kind of muddy, bloody portal vein. You would have the bright red hepatic arteries, and you would have had the green of the bile duct. All right. So that blood flow into, let's see, central vein, that's draining out. Okay, portal. So the blood flow coming in is delivered by branches of the hepatic artery, branches of the hepatic portal vein. Okay, so we're bringing in oxygenated blood, and we're bringing in kind of deoxygenated blood that we drained from all of the intestinal tract, and that's the hepatic portal vein. And we're going to send those branches in, and they're going to divide and divide and divide and encircle each lobule. Those vessels are. And by the time they get small enough to circle each microscopic lobule, we're going to call them interlobular vessels. And the hepatic artery will be down to the size of an arteriole. Sometimes we'll call that the hepatic artery branch, or we'll call it the interlobular hepatic arteriole. It's the same thing. It's just a small subunit off of that hepatic artery. The same with the portal vein. We'll call it a portal vein branch in that interlobular area, or we might call it interlobular, and that's I-N-T-E-R, hepatic venule, because it's that small at that point. We'll also have an interlobular bile duct, and we'll know it because it's going to be lined with a cuboidal, a simple cuboidal epithelium. And we're going to look at the flow of the bile. It starts with canaliculi, goes to bile ductules, which we won't really see, goes to interlobular bile ducts, and then on out to the individual hepatic ducts, which you might have seen draining each lobe and then converging together to form that bile duct. We're also going to see lymphatic vessels in these interlobular areas, but not always. That's why I like to call it a portal area instead of a portal triad, because triad means three. And what that refers to is the artery branch, the vein branch, and the bile duct. But you know what? There's usually a lymph vessel there. So it's more of a quadrad or a portal area. Well, what does all that look like? Okay. Oh, and one more thing. There's a central vein, and that's where all that blood drains to, in the middle of the hepatic lobule. Now, hold on, I'm gonna bail. I'm gonna jump slides, but there's madness to my method. Yes, I said that way on purpose. Definitely madness to my method. I want this slide because you can see the beautiful portal area very nicely in this particular slide. Now you'll notice, oh gosh, all that lovely connective tissue that defines each lobule that the pig did for us, nobody else is going to do that. So we're missing that. We have to fill in the gaps with our own imagination. But we can see the INTER lobular areas, and we can zoom in and we can find all those branches we just talked about. So these are little muscular, smooth muscle walls here to these vessels. Not much in the way of smooth muscle here, just kind of looks like endothelium, but there's red blood cells in here. Here we've got endothelium lined spaces with a proteinaceous fluid in there. Here we've got red blood cells in here, and that looks like just endothelium, not much smooth muscle. And then here we've got a simple cuboidal to a simple columnar epithelium. Well, let's logic all this out. This is a branch of the hepatic artery. It's the interlobular hepatic arteriole. This is a branch of the portal vein. So this is an interlobular hepatic portal venule. This is a lymphatic vessel. This is just some lymph in it, one side of it. This is the rest of the lymphatic vessel here. This is more of the venule. Here's a little bit of, this is a bile ductule right here. You don't usually see these, they're small. You usually only see the interlobular bile duct right here. So ductule is something smaller. So I told you probably wouldn't see it, and then there it is waving at me saying, ha ha, here I am. So there's the interlobular ductule and then the duct. Don't worry about those ductules, they're rare, and it's just there to make a fool of me today. So you'll just be IDing the interlobular bile duct right there, okay? So now let's go back to Mr. Pig. And again, if I'm going too fast, just fuss at me, because I forget. I get kind of excited about things here. All right, so here's Mr. Pig, and we can see our lobules outlined. Now we found everything on that list except for the central vein. Well, okay, it's central, so let's look in the middle of the lobule. There it is. Folks, it doesn't look like a vein. It's just kind of plain, lined with some endothelium. All right, so that's your central vein. Because the sectioning artifact, it won't always show up inside each lobule. And if it does, it may not always be central. It may be offset because of oblique. Okay, it's a central vein, but it's not central. Oh, it says my internet connection is unstable. That's cute. Okay. All right. So here is just a really expanded longitudinal view of that portal area, as is this right here. So if you see the bile duct, the interlobular hepatic arterioles, the interlobular portal vein branches. These are just really big branches coming in, and they're going to divide and send their branches out around each lobule in that interlobular connective tissue. Now, I don't know why, but when you're looking at a liver, you often only see the structures in the interlobular connective tissue in these little portal areas. There's the arterial, the bile ductal, the vein, and the lymph, might be lymphatic. I don't know, that looks like red blood cells there. It's not common for you to get to see those vessels as they travel all the way around the lobule, but they do. They travel all the way around. They have to, because they're supplying blood through the hepatic sinusoids down to, that's draining into the central vein. Now, I'm going to jump slides again because this one will let you see the sinusoids, which is what the vessels in the portal areas. Okay, here's a portal area. The poor vein got destroyed by the knife, but here's the artery branch, and there's the bile duct branch. Now, these send their branches all through the interlobular connective tissue, and they deliver blood into the sinusoidal capillaries. And here in this H&E, you can really appreciate the beauty of the sinusoidal capillaries. Sinusoids are just sinuses. You know, we've seen coronary sinuses, and we should know now that a sinus is a dilation. We saw aortic sinuses, coronary sinuses. Gosh, there's some other sinus in the heart. Sinus means a dilated vessel. These are dilated capillaries. So these are hepatic sinusoidal capillaries. And we're going to percolate that blood from the interlobular area down through these sinusoids, bathing these hepatocytes on two of their surfaces. And that blood is going to percolate down into that central vein and drain out of that central vein. While this blood percolates through the sinusoids, it's going to be exposed to the hepatocytes. Now, what do those hepatocytes do? Well, here we are in the sinusoids. And it says they're capillaries with large diameters. They're lined by an endothelium, the lining cells, sinusoidal lining cells. There's a perisinusoidal space, or the space of disc. We're not going to see that. And we need an electron microscope to see that. But that little gap between the sinusoidal endothelium and the hepatocytes, that's where the microvillous surface of the hepatocytes is, and we won't see that either. These sinusoids are going to have a discontinuous endothelium to let that blood plasma, but not the red blood cells, get through and bathe that microvillous surface of the hepatocytes so that they can do what they do to the plasma. There are stellate macrophages. Stellate macrophages, also known as Kupffer cells. Stellate means just star-shaped. There's hepatic macrophages in these sinusoids, and these are permanent macrophages. They're not climbing around. So here's a hepatic macrophage in the sinusoid. Here's a sinusoidal endothelial cell. You can kind of see the perisinusoidal space right there, that gap right there between the nucleus and the hepatocyte. That's probably endothelial cell, endothelial cell. Oh, look at this hepatocyte, isn't he cute? He's got two nuclei, so he's binucleate. Is that a bad thing? No, that's just something hepatocytes do. Is this a binucleate cell? Nope. There's a little canaliculus. So that's just a nuclei of two separate hepatocytes being next to each other. But that's a binucleate cell. Binucleate cells, the nuclei are usually, they're like conjoined twins. They're usually touching each other. They're real close. Okay. So back to the sinusoid, lined with endothelium, got blood percolating through it. You may see, look at this, granulocytes and agranulocytes. So these are white blood cells in the blood. And then macrophages are just everywhere. Here's a really pretty one right here. Very typical, right there. That's a hepatic macrophage, Kupffer cells. Some of these sinusoids looks like blood you can hardly even get through because of all these macrophages. So these guys are looking for foreign antigens that they can phagocytize because, you know, this is kind of half of it's dirty blood coming in here that just came from the digestive tube. And those macrophages have to clean that blood up, as do the hepatocytes. Let's see, biocanaliculi, sometimes they show up, sometimes they don't. So these canaliculi are little gaps, intercellular gaps in between individual hepatocytes. And that's the beginning of the bile drainage. Bile is released into these canaliculi. They converge and form the bile ductules. Remember the little guy I said that was making a fool of me because I said you wouldn't see him. And then those ductules converge to form the bile ducts, the interlobular bile ducts, which converge to form the hepatic ducts, which you can see in gross anatomy. All right, what do we have left? We probably need to go back. This is just showing you the canaliculi, the bile ductules, the interlobular bile ducts, and then the larger bile ducts. We probably need to go back to the central veins, okay? So we've delivered blood to the interlobular area. We've percolated it through the sinusoids. Now it arrives at the central vein, and what happens after that? Central veins are drained by sublobular veins. So this is a central vein, and those central veins converge to form a sublobular vein. This is a sublobular vein. Well, how do I know that? Well, it doesn't have any friends. It's no partners. There's not a branch of the portal vein here. There's not a branch of the hepatic artery. There's no bile ductule. So he's a loner and he's big. So I know that that's a sublobular vein. And the sublobular veins converge to form the hepatic veins. And the hepatic veins drain each lobe and into the caudal vena cava, okay, which we've seen, the caudal vena cava. Open up your caudal vena cava and look at all these hepatic veins emptying into that caudal vena cava. All right, the classic hepatic lobule here in the 3 possible functional units, the only one that you'll see is with the pig liver, and that was that classic hepatic lobule right here. The other 2, portal lobules and hepatic acinus, we won't be able to see. They're not defined by connective tissue, okay? But there's a good description of those. The lobule defines the bile duct flow, which is it's reverse flow. So blood is coming from the interlobular area down to the central vein. Bile is flowing opposite that out to collect into the bile ductules. And then the acinus describes blood flow in a manner that pathologists use, because you get more toxins delivered in this outer zone, less toxins here as the hepatocytes detoxify toxins, and less still yet here. So the amount of toxins decreases as you get towards the central vein, but the amount of oxygen decreases as you get towards the central vein. So it's kind of yin and yang between toxins and lack of oxygen, and pathologists will talk to you about that. But that's why we introduce you this liver acinus concept, because you'll get that concept again in pathology when they talk about toxic insults or lack of oxygen to the liver and what it does to the hepatocytes. Let's see, there was a mention in here about storage of glycogen and lipids. And I thought I had it highlighted somewhere. Yeah. Some differences can be ID'd without special stains, such as glycogen and fat spaces. So I wanted to show you that. We'll go back to Mr. Pig. We'll zoom in to a classic globule. Even at this level of magnification, I can see that the outer, maybe about a half of this lobule looks grainy and different than this inner half of the lobule. Okay, why is that? Well, the hepatocytes store lipids and they store glycogen granules. So this moth-eaten grainy outer portion that's closer to the interlobular area, this is grainy pattern is mostly due to intracytoplasmic glycogen stores. But as you get closer down to the central vein, you tend to get just more like round, clear vacuoles. That would be more like lipid storage, little intracytoplasmic lipid vacuoles. Okay, so that's a couple of staining characteristics that you can distinguish with light microscope in these hepatocytes. Again, the grainy glycogen storage spaces, and then the more spherical and larger lipid storage spaces that are closer to the central vein. Okay. Now, last but not least is the gallbladder. And you met Mr. Gallbladder in Gross Anatomy. And here it is on this H&E slide of the liver. This is a canine liver, by the way. And this is the gallbladder. And so what was it attached to? It was the right medial lobe. Okay. The right medial lobe is where that quadrate, where that gallbladder was attached. It also kind of squished up into the quadrate lobe, but it's attached here, fused to that right medial lobe right here. And you'd have to kind of peel that gallbladder out of that little fossa where it resides. So this is probably a section of canine right medial liver lobe with a part of the wall of the gallbladder. Now, there's not a lot you need to know about the gallbladder microscopically. Let me zoom down here to where it is. Really, you just need to kind of ID the organ. How would you do that? Well, you'd need to see it with a piece of liver like we have right here, or else we'd have to show you or tell you what it is. And really, all that we kind of have you do is ID the epithelium that lines the gallbladder. We just don't get too excited about the gallbladder. Note that these are not glands right here. These are just mucosal folds. So that it's almost like gallbladder rugae, but they don't call it that. You know, you get folds in the mucosa of the glandular stomach when it's empty. These are mucosal folds, and we just can't see that they're actually connected to the lumen. So these aren't glands, these are mucosal folds. And this is said to be the tallest supracolumnar epithelium in the body. I don't know if I would challenge that statement or not, but it's pretty close. And these are simple columnar cells for sure, and they are pretty daggum tall right here. So very tall, simple columnar epithelial cells. That's about it. If you can tell us that about the gallbladder, we won't get too excited about it otherwise. If that's a deal with y'all, then we'll shake on that. Any other questions you have? Is there anything in the chat? Because I think I've run through everything I wanted to show you today. I knew it would take about an hour, but